This is the International AQA GCSE Certificate, and now we're going to have a look at how we can measure the distribution of organisms within a particular environment. Last video we gave consideration to the factors that might cause changes to these distribution of different species, whether they be living factors, so other animals and plants, or whether they be non-living factors, such as the introduction of or changes to temperature or pH and so on and so forth. So here we're going to look at how we actually go about taking those measurements and monitoring whether that change has actually taken place. To do this, there are going to be a number of tools that we require. The obvious ones are going to be a tape measure to start with, to set out our areas of measurement. We're also going to have a look at quadrats, and quadrats can be either open or gridded, are the two main types. So an open quadrat is quite simply a frame of four sides, and it is ½ a metre by ½ a metre each way round, giving you a total area of 0.25 metres squared. Every quadrat is always the same because that gives us standardisation to everything that we do. It's also been calculated that that's the best size for it to be in terms of the optimising time taken to carry out every sample, but also the accuracy of the results that you get. So that's an open quadrat. If we were to use a gridded quadrat, it would be just the same size, but it would have, as its name would suggest, a series of grids all the way along it, and there are 10 grids that way and 10 grids that way, giving you obviously therefore a total of 100 squares. These gridded quadrats are much more useful for looking at percentage cover. So plant life or something like that that's too many to count or difficult to count, you work as a percentage cover, and because there's 100 squares you can count the number of squares and easily get a percentage rather from that. Whereas an open quadrat is usually done for the numbers of organisms. So for example, the total number of snails that you find in that particular thing, that's quite easy to count. You just quickly go through the whole quadrat without it being underneath a grid that's hard to get through. We also therefore would need our abiotic tools, so a thermometer, a pH probe, or whatever data we decided we were particularly going to collect on that occasion. We can also have to consider exactly what it is we need to design our experiment with. If we've got a really big area that's going to take a long time, so how much of it do we want to cover? These are the other sorts of things that you might have to think about when planning an investigation. You would have hopefully come up with an idea about what you think is causing a change, so you'll set out to go and measure that. So maybe temperature, maybe light intensity. That might be what you think, in your hypothesis, is affecting the distribution of your organism that you're measuring. So you need to go and collect that. How much do you want to collect? Are you going to do lots and lots of readings? That improves reliability, but it could also increase the time taken to measure it. It might not be feasible, certainly if you're on a beach and the tide is coming in. What time of day are you going to do it? What time of year are you going to do it? And if you're going to actually make meaningful data, do you need to come back over the course of a year or a week, different times of day, whatever it may be, to get the best, most reliable results? That might not be practical, and that's just a flaw in the experiment that you have to just accommodate. Key things also when designing your experiments and limitations you might come across in your exams are, did the same person do the measuring? Some things are slightly subjective, and people won't do things the same way, so it's important in your group that the same individual was responsible for measuring the height of something, so they always did it the same way for them, for their way of doing it. Particularly if it's identifying organisms off a key, like a plant key or something like that. Make sure that the same person uses the same thing, so they're always looking at the same organisms, calling it the same thing. Every other factor must be kept as constant as possible. Not always possible. It's not always easy to do that, but that every effort should be made to do that. And when you're evaluating an experiment, these are the sorts of things that you should be giving consideration to. Rightly or wrongly, they may not just be able to be controlled. So there are two key techniques that you need to know about for measuring distribution in an area. And the first one is random sampling. And this is the sort of thing you would do if you were covering a really, really big area and you wanted to gain just an impression of what is the distribution of the species within that area. So a really big field, and you were asked to go and tell me how many ants there were there. It's impossible to dig down and get an idea for every square centimetre of that field. So you would use this technique, random sampling. And to do this, and you've done it yourselves, we have randomly generated coordinates, these points within the area, and the reason they're random is so as to avoid bias. So you don't look at an area and make sure, I know there's a good set of ants over there and a good set there, I'll make sure I go and measure those, because that might not be a fair distribution of the ants in that field. They might just be the clusters, the colonies of them. So avoiding bias by you having any form of choice as to where those measurements are taken is massively important. So to have a look at how we would do some of this random sampling, if this was your field, you would set out your tape measures like a set of axes, and your randomly generated coordinates from a computer, not just picked out of your head, randomly generated would give you coordinates. So you might have 6, 6 and 8, 2 and so on and so forth. They could be your meters, so you have your x-axis and your y-axis. So the first one, I'd come along 6, up 6, I would place my quadrat down there. Similarly, I'd go along to 8 and up 2 and place my quadrat there. So these locations to put the quadrat are randomly selected. And it may well be, just because of the chance of the coordinates that I collected, that they happen to all be there in one area and ignores a patch of the field. If you haven't got time, then that's a flaw of the investigation. If you do have time, you can increase the number of quadrats that you take, generated from your random numbers, so as to give you a greater chance of encompassing all of the field. So hopefully you might get some more areas like that. Similarly, you might get them all in one area, but then certainly not going to be evenly distributed, which is probably what would happen if a person was to choose precisely where to put them. So the coordinates are random, the quadrats are random, and within your quadrat then obviously you count the number of organisms, measure percentage cover, measure your abiotic data that you're looking at as well, so light intensity or whatever it may be, and you can collect your data from there. So that is random sampling. The next technique might be to look at taking a belt or line transect. This is so as to give you a much better idea of how the distribution of an organism changes as it moves through a particular habitat. So to go back to our area, having got a general idea, we want to see, well, why does one population cluster here and not there? So let's see what changes within that environment there are as you go from left to right that might be the cause for that. So to put a transect on, we would get our tape measure this time and set that as a straight line all the way through the middle of your quadrat, between point A and B, for example. If I just quickly redraw. So here's your transect going through, and this time we actually put our quadrat down at regular intervals. Rather than random, they're regularly spaced. So this is a systematic approach. Now, These can be spaced at any interval you choose, and it's dependent upon the size of area that we're working in, how long we've got to do it. So it may be that they are at 2 metre intervals, or they could be at 5 metre, or they could be at 100 metre intervals. It just depends on what it is you're trying to achieve and the size area that you're working within. And at each of these places, not only do we, again, measure the number of species that we're interested in monitoring, we would also measure our abiotic data, the light intensity, the soil pH, whatever it might be. And hopefully, from there, we should get a much clearer idea of how our environment has changed as you move across your habitat, as well as how the distribution of your particular species has also changed. And then we can come to analyse the data. And so there are key things that we have to consider when looking at that as well. How many organisms are there? You usually do that through some sort of a tally chart or identification as you're out in the field. But of your species, and of the data you collect, you have to do some calculations. You need to know about how to calculate on what a mean value is, which is the average value. The median, which is the middle value. Certainly if you're going across a field, you want to know what is your middle value of number of species. Maybe that would be the most common one. And then mode is the most common value. So how many times does, say, number 6 appear? Okay, so these two are slightly different. The median is the middle value. The mode is the one that appears most frequently. And you would have to be able to identify those from a set of data that was given to you in an exam question. Once again, there's a strong emphasis on evaluation of this as a practical skill for your exams. So evaluate always means give the pros and cons. They often give you data, or they might give you an experiment, and you've got to say what was good and what is bad about it, and sometimes give a conclusion as well. That gets you a separate mark. So, have you taken enough samples to trust the data? Has there enough abiotic data for it to have had an impact on the distribution of your species? Have you repeated the whole process enough times? Often, the number of samples and number of repeats are often tallied together in some investigations. Have you done it at a particular time of year? Should you have come back at different times? Did you choose the wrong time of day? Anything like that. Identifying is usually a key error. If people change, we talked about before, they identify things slightly differently, they interpret data incorrectly. So those are the things to try and keep the same. But they also ask you to give suggestions, and that's not only suggestions of how to improve on your evaluation, but also suggestions as to possibly why you found what you did going through your areas. So they'll give you data, they'll see that the population is decreasing if you go from one side to the other, but perhaps you have to suggest why. What else have they given you in the question, or is there in there that explains why the pH of the soil may have dropped? Why is it that the light intensity has dropped? It could be because there's a shadow from the building, for example. You've got to give suggestions as to why the results have done what they do. Thinking about the biology elements, obviously. So light intensity, passing traffic, trampling of people because there's a footpath going through a particular area. Maybe if somebody introduced new predators, maybe it's closer to the water so there's more food available for certain species. Give sensible suggestions as to why those organisms may have distributed themselves the way that you find that they have. What would be really important as a useful thing to do might be actually to go to your notes, and on the last page of your notes here are some possible suggestions of different investigations that could be given to you. You've done some of them, others could be given to you in an exam question. So you should go in and look at these and try and consider what type of technique would you use, the belt transect or the random sampling technique. Why is one better than the other for this aim of investigation? What tools do you therefore need to do that? And what sort of data could you get, and what graph might you draw? A bar chart, a line graph, a scatter graph, whatever. What's going to be the best way to interpret that data? And then obviously suggest some reasons as to why you think changes might change. So limpets and barnacles, what are their risks? Look them up if you don't know what they are. Look at some pictures. They're living on the rocky shore. They're exposed to the wind and the waves and the sun, etc. So where are they going to be best suited for living and worse suited? So have a look at some of these and practice coming up with some evaluation answers and some technique descriptions as to how you might actually measure that data.